Good morning, professors and all researchers. My name is Chen Yang Tong, the student from the Institute of Applied Mechanics, National Taiwan University. Today, the topic I want to report, that is, electricity and instrument-free infectious disease sensor based on multidimensional origami paper-based analytical device. This is the outline. In this report, I will introduce HIV and infection in the world, point-of-care tests, which is made by WHO, timer paper-based analytical devices, HIV-1 P24 detection and conclusion. According to WHO Global Progress Report on HIV, viral hepatitis and sexually transmitted infections 2021, there were approximately 37.7 million people living with HIV at the end of 2020, with 1.5 million people becoming newly infected in 2020 globally. The main cases are more likely to occur in lower resource areas. It is one of the top 10 causes of death. Hence, HIV testing is a very important issue in the world. WHO has established several standards that are suitable for testing in lower resource areas. The name is point of care. Its topics are affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable. According to the concept of point-of-care tests, we designed a device which called timer paper-based analytical devices, TPADs. Magnets and 3D printed cartridge are used to tightly combine 3D paper-based analytical device. The method not only effectively reduces the gap between the papers, but also allows the reagents to flow uniformly and stably in the paper microchannels, which reduces the possibilities of detection errors, external interference, and non-specific attachment. The entire platform is divided into two parts. The left two wells are control well and test well. The right two wells are the timer, first and second light on, to indicate washing and TMB reaction finished respectively. Before the testing, we pre-treat the T-pads to achieve the purpose of user-friendly. The first area is comprised of a detection pad featuring test and control detection wells, which are surface modified with capture antibodies used to absorb the antigens when the sample solution is added onto these wells. The secondary area includes a strip of paper featuring two conjugate wells with dry detection antibodies, which are washed down to the detection wells after adding the washing buffer to the conjugate wells, and bind with the antigens absorbed by the capture antibody on the detection wells. To incorporate an ultimate timer function in the device, we deliver the excess washing buffer from absorbent pads to the dye wells, which are pre-treated with dry green food dye. By pre-treating the paper substrate with dry sucrose and purulent, we are able to control the viscosity and therefore the flow rate of washing buffer to ensure it transfers the dye to the indication wells at the appropriate time, thus coloring the solution and turning the indication wells green. Based on the origami design of the purpose 3D T-pad and the developed timer, the operation of the detector can be simplified into 4 steps. 1. Sampling. First, we add the test sample to the T well and control sample to the C well, which have been pre-modified with capture antibodies. After adding the samples, the antigens contained in the test sample will bind with the capture antibodies and form the antigen capture antibody complex on the T well. 2. Washing. Next, we pull down the paper strip until the T and C wells overlap with conjugate wells, to which we add 100 microliter of washing buffer. The HRP conjugate detection antibodies pre-dried on the conjugate wells will be rinsed onto the T and C wells after adding the washing buffer and react with antigen antibody complex formed in step 1 on the T well. In addition to rinsing the antibodies, the addition of the washing buffer also activates the timer. Specifically, after being added onto the T and C wells, the excessive washing buffer soaks into the underlying absorbent pads, where it dissolves the pre-dried sucrose and purine, and then being colored with green after melting the pre-dried green food dye on the dye wells, and ultimately delivering the green color to the indication wells to indicate the timing for performing the immunoassay steps. 3. Color development. When indication well 1 shows green color, we slide down the strip and add TMB solution to the T and C wells through the holes on tab 3. As a result, when the biomarker is present, the T well will display a dark blue color due to the reaction between the TMB solution and HRP tag on the detection antibody, while the C well will display a light blue color due to the reaction between TMB solution and HRP conjugate on the residual detection antibody that physically absorbed on the C well. 4. Comparing. When indication well 2 displays green color, this indicates the optimal TMB and HRP reaction time. At this point in the test, the target analyte concentration can be determined by comparing the color on the T well with the color chart diagram. To demonstrate the feasibility of the purpose origami-based 3D T-PASS design, we used the device to detect HIV. We chose the viral capsid protein HIV1 P24 as the target antigen. Using the purpose sensor, we established the standard curve of the HIV1 P24 protein concentration. We found the BR values increase as we increase the HIV1 P24 protein concentration. To validate the feasibility of the purpose 3D T-PASS in detection clinical samples, we further performed the tests with human plasma spiked with 0 nanogram per milliliter to 30 nanogram per milliliter of HIV1 P24 protein. The resulting BR values of the detection wells increased as we increased the HIV1 P24 protein concentration in the plasma. The linear detection range was from 0.03 nanogram per milliliter to 3 nanogram per milliliter with an R-square of 0.97 and the detection limit was 0.01 nanogram per milliliter. This is the conclusion. In this study, we integrate the timer into origami analysis platform, replacing the electronic timer successfully, and we use CMC EDC NHS for surface modification. The capture antibody can be more firmly attached to the paper, replacing expensive glass or nitrocellulose film and increasing its sensitivity. The detection limit of P24 and P24 which spiked in plasma is 0.01 nanogram per milliliter, and the detection result can be visualized after 5 minutes. This is the end of my report. Thanks for your attention.